I've been introduced. My name is Bettina of InnoVitro. We are CRO. We are a contract research organization. So we provide or deliver services on human-based iPSC-derived cell models. And we are in the cardiac risk assessment area active. And we had the question whether some of you want to show some muscle cells, so I can present them to you and we'll tell you a little bit as to how we have developed this company. And since we do not only offer the assay system, but we also co-developed it, and this has to do with the technical research area. And since we are a service provider, I'll bring along 2 case studies to give you an impression as to what kind of issues and questions we'll deal with, what we can test by means of our assays. So here we have got this kind of key method with which we've worked. It's a 96 well-played system and ethically very correctly developed human-based services for drug development. And here you can see it in the video. It's one of the precursors of the first few models we had during university time in 2001, where the cells, as you can see in 2011, you can see that on the timeline, where we had the first cells and placed them there, and we saw people are interested in it, but you have 6 wells, and you can't get very far. If you want to industrialize it, we have to get further, and that is when we set up the company. This is Matthias, our CEO, Peter, our CTO, Jan, our lab manager, and myself. This is a core team, which still continues to exist for many years. We cooperated with NATworks. It's also a German company. Some electrophysiologists well know it. They determine the appliances, and we together developed the FlexSite Nixis 96 tech system together with a membrane, and I take the heart cells because they're beating so clearly. In a cell-based assay system, you normally don't see a lot if things are not moving. But here you can see them with your eyes because they're moving cells. But we not only reflected or referring to this to these cells, but we used also other technologies. This was only the starting point to round up everything, and besides the continuity in this FlexSite 96 area, we also deal with electrophysiology, and we also do live cell analytics, so that we can offer a whole package to our customers. If we compare it to what existed before at animal level, so we have the typical Langendorf heart, which was used where you use rodents or frogs. The species is not the same, but of course this may lead to infections we don't see in human beings. You have a very low throughput. 1 heart, 1 animal, 1 experiment. But you see the whole organ, it's very physiological. But here we've also got the physiological relevance because we have the membrane. And with a view to stretchness, we focused on the heart, a heart type of membrane, very close to the heart. Because of the FlexSite 96 throughput, we have very good experiments, and the flex-based model is really an asset. But perhaps I can add a little bit on it, what the failure rates are at the moment in the job development. There are different phases and different failure rates. This is from a paper dated 2013. A lot of things have changed, not for the better, I'm afraid. And from the marketing area, you can see that even drugs had to be withdrawn from the market. If you look at the organ systems, which led to first drug reactions and attritions, so the withdrawal of drugs, what are the reasons behind it? The problems in the field of the cardiovascular system. That's the blue part, marked here in blue. And one of the reasons, because it often leads to problems in the market, so there were arrhythmia problems which could not been foreseen by the kind of testing used at that time. But in 2010 and 2011, the CEPA initiative was born because we wanted to try to improve this by a new approach, so that you have the SQT, elongation, extension, and there are different blocks, as you can see here. We have got one-channel testing, but then it was added that in silico, where it was inserted also in clinical phases, people aimed at improvement. But what I'm aiming at, we wanted to have human-derived stem cells, cardiomyocytes, that want to be tested. And I'll come back to this in a minute. These are the cells we use in our system. As you can see here, like in the genetic model, we still have the drawback. It's good that we have the human iPSC-derived heart cells, but on the other hand, they are very young in their phenotypes. And if you just think the most compounds or drugs are meant for an adult person, this isn't what we want, because a young cell doesn't react in the same way as an adult cell. And there's a lot of information available as to what leads to improvement of these cells. We have the prolonged culture times. If you think how long a heart in the human being takes in developing until it's regarded as a human heart, hormones, extracellular matrix, CT, 2D, and the substance stiffness. The normal area we normally have, and most of you will know, we have a cell culture as plastics or made of glass, and this does correspond to what the cell finds in a body. And this is where we come in. And that's my favorite video, because here you can see the membrane perfectly. With the membrane, it's possible to have a look at this effect, give it a better environment, and to give it opportunity to mature in vitro. The technology works as follows. In the lower part, you can see that we have got electrodes, but they're not connected to the plate, but they belong to the device as such, the appliance. And when the cells contract on the membrane, they pull up the membrane. And between electrode and the membrane, we can measure with capacity distance measurements. And this shows the contraction force, which can be defined and measured, and other parameters as well. Most of you will know this as well. And we have the amplitude, the contraction force we can measure. This is just a single contraction, what you can see here. And we also have to determine the arrhythmias or the beat rate. That's here the early after contraction with people that are active in early after depolarization. This is what from the electrophysiological level is transported into the physiological movement. But we may also determine the slopes, the breadth of contraction, for which is good for QT extension, and the whole surface of the contraction cycle. And since most of the data I'm going to show you have been evaluated, I love to show you the raw data. This is the data record we've got. It's a 30-second cycle we record. This is the beat rate. the regular beat rate and of, well, then you can see here non-regular beat rate, as you can see here. Here you can see the differences compared to the normal, the ordinary one. Then we have the early after contractions. I mentioned before these after contractions, these small kind of bumps you can see down here. Or you go to jubilation and see the cells aren't well off. And that's very important to bear in mind with a view to the juvenile heart cells. They often have the problem that they don't react to the typical testing substances. Now we have got a wonderful physiological model. Now we must be able to show that the cells are get riper and mature. We leave them in a culture for 1 week, and we take the test substances. We take substances which have an effect on the force. They have an inotropic effect, and you can see the bump here that the contraction gets strong and stronger depending on the concentration. We see it for isoprotonol and for assay and for omnicomtive mecabil. They react as they should, and if we do further analysis, we do not only have a mean beat from one suite, but we can have an individual look at the elements up structuration, down structuration, which helps us to differentiate between contraction and the part of the relaxation of the cell, and we can then interfere and see what happens. This takes me directly to our case study, because the inquiries come in, I have problem XYZ, can you solve it for us? And this was the screening of substances which have an effect on cardiac myosin. This was a pharmaceutical company that was in an early stage of drug development, and the earlier the more compounds, a lot of compounds, 1,300, and they wanted to see how quickly we can do it. Well, with our 96 system, we are good, but there are higher systems available, but not in a physiological system. I think here we fare quite well in this area. So we wanted to know how to go about it. We said, okay, this is our study design. We take HIPSC cardio cells, ISLCM, the beat rate and the contraction force and the beat duration. And of course, we have to concentrate on the parameters, and we do that in an acute way, about 100 compounds in concentration as we want, replicates of 4. Well, that was well received, and what we normally do when it's possible, we have use our internal kind of book, and which are not relevant, but we can compile all the data and see what kind of compounds we have which react on cardiac myosin. We have got 3, which I brought together here for you. And we can say for mevacantem and plebiscitine, both myosin inhibitors, so reduction of contraction force expected, and depending on the concentration of the compound. When you go very high, you can even knock out the cells. We all had omecamtiv mecabil, which was also on the other slide. It was a myosin affecting compound, an activator, and this is that we here have a stronger contraction force. We could see that in our system compounds which have an effect on cardiac myosin, and that can measure. And it's possible to map their effects. I love this one here in particular. This is a way in which we can present the parameters at the spider plot. We have so many parameters, and this is a meta overview. Of course, you can always go into detail, but for a presentation, it's very fine to have here the contraction force. We look at the beat rate and the slopes, and of course the broadening of the contraction, which can be viewed very well. So MAVA comes in here on the left-hand side, has reduction in contraction force as a consequence, and here on the right-hand side, it's reinforced. In this way, we were able to show to our client, our potential client, and we were able to convince him fully. And we got the order and offered him the services. That was a wonderful case study, which of course kept us very busy. The next case study is a typical one, although it's not what comes on the first side. The characterization of in-house cardiocytes, normally we work with the commercial ones, which are produced, they're well characterized, everything you can ask with a view to genetic background, phenotype, etc., immunostaining is well known. But with all the typical commercial providers, well, we closely cooperated and evaluated all the cell types so that we can start right from scratch and quickly. But there are a lot of biotech companies or pharmaceutical companies who want to do this in-house, and we produce our own cells. But of course, they need testing. And then this company approached and said, we have our own cell model, but we would like to have some testing. How would you go about it? So what kind of inquiries we get in terms of a lot variance, variability, so we can jump from one to the next. And what a lot of them want to know, it's prekaryocytes for research. Well, then it's with a view to SIPA study. This has become a kind of standard when the cells react in the way you expect from type of SIPA compounds, which refers to the rhythmic potential, and they have a lot of strong compounds and safe compounds. We can go through it, and this is what we offered. We can do lot-to-lot variants, and we can test the SIPA variants on 5 days and also acutely. And this is what we did. Well, this is the study design, which I've just explained. And here we've got 2 different devices for the lot-to-lot variants in Flexa, the contraction force, and for the testing of the SIPA study, CardioExcel. It's about electrophysiological parameter and their measurements. I don't show you the lot-to-lot, it would take too much time, but the SIPA study, on the right-hand side, you see all the compounds we tested, and the color code tells you which ones have a very high risk, red, and the green means no risk, and intermediate, well, they're in between. Good, but not necessarily. And our testing of cells, to see here from the color mode that the strongly arrhythmic substances they reacted to, and the same for the low risk. Which was a good result, and which we then did for the customer in a big scope and said, look, if you compare it to the data published by Grinet, where also clinical data was compiled, and data of primary cells of the human heart, you can see that it fits in nicely. Intermediates here do not exist, but they were mapped in red. But it's good. We also had a look at the chronicle analysis. Everything I showed you before was acute within an hour or 2 hours at the maximum. Then we extended the period to 5 days. And then we had to look at cancer and tyrosine kinase inhibitors and added allotinib. It's a cardio-safe drug without any effect. And even under higher concentrations, nothing happens. If you compare it to sunitinib, which is regarded as cardiotoxic, you can compare the two. You can differentiate between a dose-dependent reaction or dosage time dependency, which you can see here on the right-hand side. It's an immediate reaction. If you look at pentamidine, it's a time-dependent reaction which has to do with trafficking blocker, which trafficking blocker with a high cardiotoxic risk, and so it's dosage and time dependent. This brings me to the end of case study 2. We had a wonderful study where we were able to show the customer your cells are at the level of commercial cell types. They react exactly as they should, and they react at the chronic level as expected. So we supplied good data, which they welcomed, and then now use them in-house and continue their data. I'm through with everything now, and thank you very much. I hope that to see you all again in the breakout room where we can talk more in more detail about the cells. And thank you, Tamara and Julia. Thank you also for your invitation, and I'm happy to see you every time here at the conferences. And thanks to my team, which produces the data. Thank you very much, Bettina, for this wonderful presentation.